So I presented earlier this morning on biologicals, and this afternoon's presentation's a little bit different. It's going to be more general. This morning I spoke on more on biological seed treatments, but I'm going to start out with the same story. So if you heard the story this morning, I apologize. But I've been attending these conservation tillage conferences for about 6 or 7 years since I joined 3 Bar Biologics. And 6 or 7 years ago, we had a booth down in the cafeteria, and we brought our product in, which at the time we were developing a phosphorus solubilizing product, including microbes, so living microbes. And we had some good conversations, but there's a little bit of curiosity, but a lot of open skepticism to biologicals. So it made for some interesting conversations, sometimes some rough conversations. But what I've seen in having worked in this industry now for going on 7 years is there's certainly, particularly over the last couple years, there's like a light switch that's gone on. And one of the things, after attending the conference, I told this story this morning, I'd at some point run into Randall Reeder, and I don't see Randall here yet. He'll show up with the camera. But I'd ask Randall, I'd say, hey, is there an opportunity to talk about biologicals at conservation tillage? And so 7 years later, here I am. So it took that long. But what we're seeing certainly is this, I'd say this momentum building, this interest. Okay. Certainly an interest by growers like yourselves in industry. We've seen more products make it to the market. We're just seeing a lot of activity, both with small companies as well as your major multinationals. But I think the challenge as a grower for you guys is trying to understand all of this, right? So my goal today here is to inform, educate the best that I can, being a kind of representative of the industry. And so I do have, I guess we'll advance here. What I hope you guys can leave with today is at least a better understanding in a couple key areas. So I've broken the talk down into kind of 3 segments. One is just to start with what are biologicals? What are they? And then how do you use them? So how do we apply these new technologies in the field? And then look at some field data to understand how they're working and hopefully support, do they work? Okay. So let's start with what biologicals are. And I know this is a lot and it's kind of hard to see far away, but this is, I think, a good diagram that starts to organize at least how we look at it in the industry in terms of different market segments. And what you'll see, this is going to be challenging, but hopefully I can do it. It doesn't show up at all, does it? Okay, never mind. What you'll see on the right side there is, there's basically 3 market segments. Biological control on the right side, and then on the left side, biostimulants and biofertilizers. That's kind of how we organize. I think biological control, most people understand, right? You're trying to control or deter a pest or pathogen. And there's been products on the market for a long time. Probably one of the best known is Bacillus thuringiensis, or BT, which is a known bioinsecticide that's used in a lot of applications. On the right, with biostimulants and biofertilizers, this gets into an area that's a little bit less known, right? And these products typically involve, or they do involve, handling abiotic stresses, helping with nutrient uptake and nutrient use efficiency. And so I'm going to spend most of my time, actually all my time, talking about biostimulants and biofertilizers this afternoon. Are regulated by law under FIFRA. So they go through an EPA registration. And so there's a lot of work that goes into that, a lot of testing for safety, for efficacy, before these products make it to the market. Now, for biostimulants and biofertilizers, on the other hand, as of today in the United States, there is no regulatory framework that exists. And so without a regulatory framework, the bar is low in terms of what the standards are for these products. And so what we've seen over a number of years is the impact of not having a regulatory framework there And so some products that may be on the market may not work very well. And I think you guys have a term for that. Snake oil, thank you. Some may use the term foo-foo dust, but snake oil is the one. And so that's really kind of given us a black eye in this industry, trying to get past those types of judgments on these products. But what we're seeing is more and more technology. There's been a lot of work in research and development, and we're seeing these better and better products make it to the market. Now what we need is a better regulatory process. And so it's not advancing. There we go. And so industry has spent a number of years working with government partners and officials to work towards having a regulatory framework. Obviously, nothing happens overnight, especially when you're working with the government. But where we got last year, which was a huge, huge deal, was a bipartisan effort where we now have the Plant Biostimulant Act. And so what that act does is, by law, we are going to create a product category for biostimulants, okay? You know, recognize biostimulants, they're not fertilizers, they're not biocontrol products. So they've been living in this gray area, okay? And so now we're gonna have an actual product category and then work towards getting that regulatory process in place where we have standard definitions. I'm gonna share that definition in the next slide. And then ultimately uniform labeling. Right now these products and will continue to be registered at a state level. So each state kind of has their own way of doing it, but we're gonna try to get some uniformity around that, which will help industry, and it'll also help you as growers. All right, so the Plant Biostimulant Act, the key terms here when we define a plant biostimulant is it's either a substance, a microorganism, or a mixture thereof, okay? And I like to think of a substance being the dead part of the product. And so this can be any substance that's derived from a natural or a biological source, but it's not a living organism, whereas the microorganism is a living organism. So you have to keep it living for it to provide the benefit to the plant. These products can be applied to seeds, plants, the rhizosphere, soil, or other growth media, and they act to support a plant's natural processes independently of the biostimulant's nutrient content, including by improving nutrient availability, uptake or use, efficiency, tolerance to abiotic stress, which is key, and consequent growth, development, yield, quality or yield. Okay, so that's the full definition. It took years. It took years to get to this definition, but we're glad we have it, and we're going to move forward. All right, I'm going to walk through in the next couple of slides in defining what these different product categories are. Some you may be more familiar with than others, but when we talk about biostimulants, really the function that these different products are providing is to help the plant through abiotic stress management, right? Help with plant growth, okay? Independent of fertility or biocontrol, right? And so the first category, kind of product category, involves organic acids. Probably the main products that you're familiar with are humic and fulvic acids. And you can see the functional groups shown there, or the molecular structure there. Humic acid obviously is a lot bigger molecule than the fulvic acid. It can be soil applied, where fulvic acid, since it's a little bit smaller, can be applied either to the soil or as a foliar application because it can, with its smaller size, get into the plant cells more easily. But what that functional group, which contains a carbon and oxygen there as an ion, it helps chelate cations like calcium, aluminum, or zinc in the soil. So that's one of its functional properties. The other is it has a lot of carbon. These molecules have a lot of carbon, and so they can help feed the soil microbes. So that's another benefit of these organic acids. The next product category is amino acids. And so you can think of amino acids as kind of building blocks. They include peptides and proteins, which are actually multiple peptides together. And you can get them, derive them from plant sources, animal sources. What you're seeing being used in agriculture are mostly plant sources. So these are hydrolysates from plant material that are heavy in proteins and free amino acids. And so what you see is the ability to have a lot of different amino acids, depending on the complexity of that hydrolysate, that can have a multitude of different functions. So you can see, hopefully, specific amino acids, and at least through research findings, how those amino acids provide a function to the plant in terms of either mediating stress or helping with plant development and growth stimulation. So as an example, you can see the chemical structure of alanine there, and you can see down, I think 3 rows down there, that alanine is known to help protect cold weather stress. So that's how these products can be utilized for abiotic stress management. Like to note here, enzymes, while not usually called out with amino acids, they are a specific type of protein, and they do have a purpose. Certainly, enzymes are known to as catalysts to increase rates of biochemical reactions. But for phosphorus solubilization or phosphorus mineralization in the soil, there's a couple of different enzymes that are well known to help support that breakdown of phosphorus from that's held within organic matter. Phosphatase, for example, is one. Phytase is another. So anytime you see you see the ASE at the end, that's usually a sign that it's an enzyme. And so these enzymes, you're going to see more and more products. There's been a real focus in R&D on utilizing enzymes. And so I expect you'll see some products, more products hit the market in the next couple years. Extracts. These are a common product category you may be familiar with, either derived from seaweed or plant material. Seaweed extracts, as an example, have been around for a long time. They're harvested from marine environments, and they can contain a range of different macroalgae species. Plant extracts obviously can come from a range of different plant material. The thing to understand with extracts are they're just very complex in terms of the biomolecules that are present. And because of that complexity, we can't directly point to which biomolecule is impacting what function. So you tend to see very general descriptions of what these products are doing because there is such complexity in that extract material. They also can contain macronutrients, so nitrogen, phosphorus, potassium, and micronutrients depending on the extract and how it was processed. The nice thing, being so complex, because there is such a range of carbohydrates, which are sugar that can help feed the microbes, plant hormones, and amino acids, you get a lot of potential activity there to help support abiotic stress management and plant growth. Okay. And the last product category here, and which is where most of my knowledge base comes from, and there's a lot of different uses of microbials. You'll see they span both biostimulants and biofertilizers. All of biofertilizers involve a living organism. I'm going to spend a good bit of time here with microbials today. And so earlier this morning, I talked a lot about microbials as well. The thing to understand, I'll put the pictures up here real quick. Microbial products basically can be either bacteria or fungi. At least that's what most of the products on the market today involve. And with bacteria, you'll hear the term, this is more of a researchy term, but you'll hear the term plant growth promoting rhizobacteria, PGPRs. You'll also hear the term inoculant used a good bit. This kind of is a carryover from the use of bradyrhizobium as soybean inoculants. But what these bacteria and fungi do, in nature, if you went to a field and started digging around, you'd find naturally lots of different bacteria and fungi and other microorganisms and macroorganisms there. But what happens is when there's roots in the ground, right, a plant is going to kind of naturally select its microbiome, just like our bodies kind of select our microbiome. Same thing happens in nature with a plant. But what happens is, as we farm, right, we do disrupt that microbiome and what's happening through practices like tillage and the addition of fertilizers and chemicals. As a crop input, when we add back in biology, right, with microbials, these living organisms, what we're doing is we're adding back microbes with the ability to provide certain functions, like I've been talking about, right? And these functions can help benefit the plant in some specific ways. So you have to understand what you need and what you're applying to meet that need, okay? And with the rhizobacteria, these PGPRs, what they typically do is biofertilizers to start. There's some bacteria that are effective at fixing atmospheric nitrogen, so they're known as biological nitrogen fixers. Not all bacteria can do this, there's just some that can. And that's where, like with soybean, we see this symbiosis between certain rhizobia and the soybean plant that makes for nitrogen, very efficient nitrogen fixing for soybean. I mean, I think most of you are probably well aware of that. But what happens is the plants, as they have these bacteria, the plants are actually feeding that bacteria, and the microbes are producing a range of biomolecules that provide that function. So whether it's nitrogen fixing, or in the case of other microbes that help with phosphorus solubilization, those are probably the two most known functions with products available. But then they also can produce a range of other biomolecules, hormones, other enzymes and proteins that have specific effects. And probably one of the biggest other effect is with hormones is the ability to help kind of supercharge, whether it's root growth, helping with producing a bigger root mass, or certain biochemicals that can help with stress. And so this starts to get very complicated fast. I'm not going to go in details here, but understand that there is a range of functions that these microbes can provide. In the case of the arborescule mycorrhizal fungi, the AMF, these are again another product category that's been around for a while. And their main function, fungi like this will grow on the root surface of the plant and produce these hyphae that allow for an extension of the root system. So that root system has more space that it can get into, so it helps with water uptake and nutrient uptake. All right. The way that I like to look at microbes, I think of them as little chemistry factories, right? Chemistry factories that you can't see. And what makes them special is they take in food. So they need sugar, carbohydrates, amino acids, organic acids, and a lot of that's coming from the plant. And like I talked about, the plant sends these signals, right, that draws bacteria and fungi to them. But these signals, what's happening is it's a very dynamic process, because as the plant provides food through its root exudates, then the microbes produce, as an outcome, a range of biochemistry, biomolecules that come back out. And the interesting thing here is that what they produce is very complex. Okay, all these biomolecules, or another term that's used is secondary metabolites. And these molecules, it depends on the microbe and its genetic makeup and what genes are actually being turned on or turned off, depending on that signaling from the plant. But it becomes a very dynamic process that in nature happens when we don't disrupt it. But what we can do by adding these products is, with certain functions, we can have that signaling happening and provide direct benefits to the plant. And so what's important here to understand with the living component of these microbial products is when this signaling is happening and it's very, this crosstalk, if you will, between the plant and the microbes, it becomes a very effective system to help, particularly with abiotic stress and helping with plant health outcomes. All right, the next couple of slides I'll get into like how this works in a couple cases with biofertilizers, and I think Nitrogen fixation is a big one. I think that's where a lot of focus, certainly in product development, that's where a lot of focus has been in getting more microbial products to the market that can do biological nitrogen fixation. And what I hope you can understand from the process of fixing nitrogen is first of all, it's a very energy intensive process. So to fix nitrogen from the air, it requires ATP is a fancy term for the energy that's needed at a cellular level. What's involved in that ATP is phosphorus. That's part of what the P stands for. So you need phosphorus available to provide the energy that's needed to fix nitrogen, okay? You also need an enzyme called nitrogenase. So again, remember, anytime an enzyme's present, it's going to have an ASE in the end. So nitrogenase is the enzyme. It actually gets inhibited by oxygen, the presence of oxygen. So if you have a soil that has it's very arable, has a lot of oxygen or air, that can be a problem for fixing nitrogen because the enzyme can be inhibited. Okay, so that's one thing. And then the other is, yeah. So the thing with phosphorus is you need to have enough available phosphorus for the energy needs. So it's a system. And I don't know if I have the data in this slide deck, but where nitrogen fixers can work better is if you deliver a nitrogen fixer and a phosphorus solubilizer together, there's been shown better, I guess, greater impact overall than if you deliver them separately, right? So that's where, you start to think of what the needs are, and then you got to understand what your soil can provide, right? And so phosphorus availability and kind of what level you're at. If there's a lot bound up, that's where applying something with a nitrogen fixer to help make phosphorus more available can be useful. But that's all being driven by that energy demand for the process, right? That there is available phosphorus there to support the reaction. Oxygen can be a problem, like I was saying. And then if there's a lot of nitrogen already there, since it is such an energy-intensive process for these microbes, they're lazy. They're not going to go fix more because they already have enough, right? It's only when there's a limit there, there's not enough nitrogen there, that they're going to start to actively fix, okay? So those are just some things to keep in mind. The list of bacteria there is not exhaustive. There's more nitrogen-fixing bacteria. They do fall into this category known as the diazotrophs. That's a term used to say they're bacteria that can fix nitrogen. The most commonly known strains, I think, have been used are Bradyrhizobium and Rhizobium because they form this very specific association with soybeans, so this endosymbiotic relationship, which makes nitrogen fixing very efficient for these strains with soybean. When we talk corn, we don't have that same sort of symbiosis relationship between a microbe and corn. So nitrogen fixation is not as efficient with the other strains, with strains that can fix nitrogen, but they're free-living or they're endophytic. So free-living microbes, they will associate with the plant root system at a certain level, but they're free-living. Endophytic microbes will actually get inside plant cells. And so there's some new products on the market that do this. They get inside the cells and then they fix from inside the plant, fix nitrogen from the atmosphere. So some things nuances just to mention. So nitrogen fixing products, particularly used in soybean, are not new. They date back a long time. But I want to point out what is new and coming. Just some of these products you probably have seen on the market. But Bradyrhizobium and Rhizobium have been around for a long time. Other naturally occurring microbes, Azospirillum species, Azotobacter are two other common types of bacteria that are known to fix nitrogen, and there's a number of products on the market that contain them. But what we've seen lately are, I think, what is increasing this interest in this area are these new products that are coming on the market. And this is an example of a few that have become available in the last couple years. The I don't have my pointer here, but the first one there, Invita, this includes one of those endophytic microbes that I was talking about that fixes, it gets inside the plant cell and it fixes the nitrogen. So it has a little different way of doing it than like an azospirillum or an azotobacter. Eutricia is a product, this is a methylobacterium, it's another endophytic microbe. It actually, it was developed by a company in Spain, Symborg, and they first commercialized it as Blue N, and then Corteva brought it over here to the US and has been selling it as Eutricia. Corteva just acquired Symborg. There's a lot of mergers and acquisitions happening in our industry right now with some of these startup companies that develop these new technologies, and then they end up being with one of the big global ag companies. So this is an example of that. Invita, the company Exotic, is based in the UK, and they've been selling Invita in the US for a couple years. So that's two examples of endophytic microbes. The next one, these are all naturally occurring microbes. They've been isolated from the environment and developed by these companies. What's coming, and partially already here, is the use of synthetic biology, okay? Whether that's, synthetic biology involves genetic engineering of some sort, whether that's by gene edit or by genetic modification of the microbe, okay? And so some of you may be familiar with Pivot's Proven product that came out a couple years ago. And Pivot's the first company to bring a gene-edited product to the market. It involves basically what they've done. I talked to you about the challenges of fixing nitrogen when there's too much nitrogen there. The microbes get lazy. What Pivot did was it created a gene edit that said, I don't care how much nitrogen's there, you're still going to fix nitrogen. And so that's what their gene edit does. It allows nitrogen to continue to be fixed even though there's nitrogen that may be present at a high level in the soil. And then the future. Ginkgo Bioworks is one company that is well known. It's a big, it is the biggest synthetic biology company based in Boston, Massachusetts. They are working across many different industries, but they have a whole division focused on agriculture, and their whole point of existence is to work on genetic modifications to improve these microbes. They're starting with nitrogen fixing, and they have other products microbes that they're working on. They have not brought a product commercially to the market yet, but they're working on it. So that's what the future holds, right? We will see more technology, better and better microbes, and it will, at some level, involve synthetic biology. It will. All right, I'm going to move on, talk about phosphorus solubilizers as another key product category within biofertilizers. And the big thing to understand with phosphorus solubilizers, depending on the phosphorus source, whether it's inorganic or organic. So the main way that microbes help to solubilize phosphorus, so in the soil, it may be bound to a range of different minerals. And what the microbes do is one of the main ways that they work on solubilizing the phosphorus is by producing a range of organic acids. And so that lowers the pH, and then that helps to kind of unbind from the phosphate that's attached to potentially calcium or aluminum, whatever the mineral is. It frees it up and makes it more bioavailable for the plant. In the case of organic sources, soil organic phosphorus, this is where enzymes come into play. So there are differences in how phosphorus availability happens in the soil. The specific enzymes involved are phosphatase and phytase. And so you may see products that either contain these enzymes or have microbes that are going to produce. And there's a range of different microbes that can do this: Bacillus, Pseudomonas, Rhizobium, penicillin. Okay. All right, we're going to move on to how to use these products and get more into the application. I talked a lot this morning in this morning session, so I think these presentations are available at some point. I talked a lot about application equipment and delivery, specifically seed treatment and furrow delivery. So that presentation, if you're interested, that goes into a good bit more depth than what I'm going to talk about here, so you might want to check that out. But if there's two things that you remember today that you can take with you, pay attention for the next minute or so. When we talk about microbes, we talk about two things primarily to keep an eye on. It's colony forming units, so CFUs, and logs, okay? CFUs. This is how we measure viability of microbes in a product, or microbes in general. So in a microbiology lab, we do a lot of plating. We can take a sample out of a product, plate it onto an agar plate like you see here, and then we'll know, based on what grows on that plate, what's there, and how much of it's there. And so we'll count the colony forming units. So CFUs. When I talk about the label in the next slide, or in 2 slides, you'll see why CFUs are so important to understand, okay? The other thing to understand with microbiology is it's exponential. We deal with logs. And sometimes that's hard to translate. We're very linear thinking, so when we have to start to think about things exponentially, it's a little harder to grasp. But the big thing here is if you think about taking, say, 1 million bacteria, right? This is 6 logs, okay? So 10 to the 6th. If we lose a log, so you think about log loss, and we go from 1 million cells to 100,000 cells, we're going from 6 logs to 5 logs. But what that translates to is we've just lost 90% of our product. Like 90% is a high number, right? 1 log doesn't sound so bad, but when you put it in terms of percentages, it sounds pretty bad. And then when you talk about losing 2 logs or 3 logs, you're essentially going from, say, 1 million or 6 logs down to, say, 4 logs after you lose 2 logs. And that ends up being 99% reduction. So you're losing 99% of your product. So as you see there in the table, with each log reduction, you're going from 90% to 99% to 99.9%. You can see how bad this can become. So when you receive a product that, at some point it had to get to your house from the manufacturer, right? It was stored, it has microbes in it. Living microbes die over time. No matter how good the formulation, some microbes have a little bit better shelf life than others, but they die over time. So you're getting log loss before you even use the product, okay? And then you're going to apply it in some way, whether a seed treatment, delivered in furrow, and then you're going to experience some more loss, okay? And then you're going to experience a little bit more loss in the field, which I'll talk about. So the idea is you want to start with as high of a concentration as you can because there will be some log loss happening over time, okay? And that's what it means when you start to look at, with each log, what you're losing in your product, okay? All right, how this translates to what we do in the field is ultimately we're going to deliver these microbes. Most of the time it's a very precise delivery as a seed treatment or in furrow delivery. And ultimately we want to get so many CFUs per seed. That's our goal. And while there are no current regulations in the United States on what that should look like, there are in other countries or regions around the world. There is some variation from a regulatory perspective, but in general, the recommendations for larger seeds like corn and soybean is to achieve 5 to 6 logs, right? Or 10 to the 5 or 10 to the 6 CFUs per seed, okay? That's what we're trying to achieve when we deliver these microbes. So let's talk about labels, because this is where it starts when you get a product, right? And some of the things to understand on a label, there should be a minimum guaranteed analysis on the label that lists the microbes that are in the product. It may be 1 microbe. It may be multiple microbes. Okay, and you see in this particular example, there are 2 microbes present. One is a Bradyrhizobium alkani strain. It has a fairly high concentration associated with it, 10 to the 10 CFUs per milliliter. Okay, so that's a liquid formulation. If it's a dry formulation, it'll be in terms of CFUs per gram. The second microbe that's included in this product is Delphtia acidivorans. And it has a lot lower concentration, basically 10 to the 7 CFUs per mil. Make sure you understand what the microbes do. So I think Bradyrhizobium, this is going to be used with soybean. It's pretty folks are fairly clear on bradyrhizobium being a nitrogen fixer. But what about the Delphtia? I've never heard of Delphtia before. What does it do? Some labels will have good indication. In this case, how it works is on their label. And at the last line of that, I know it's hard to see, says that this Delphtia strain is a plant growth promoting rhizobacteria known to enhance plant growth and yield. Okay, so it's a PGPR. It's helping with plant growth, plant health. Okay, they don't tell you anything more in terms of what it's doing, but you at least know that it's a biostimulant helping with plant health, abiotic stress. Okay, so that's important to understand. And then you need to understand how the concentration of what's there in terms of their guaranteed analysis translates to what you're delivering in the field. So that's where the application rate comes in. And you can calculate from the CFUs per mil with the application rate, you can get to number of CFUs per seed. You can see the math there. And for the bradyrhizobium strain, because it is at such a high concentration, we end up at roughly 10 to the 6 CFUs per seed based on the application rate and what they're saying is in the product. So that's good, right? The second strain, the Delphtia, when you do the math, it ends up at 3 times 10 to the 3 CFUs per seed. It's basically 1,000, 3,000 CFUs per seed. Not a lot, okay? So understanding what differences those logs make is important. Lower than the recommended 1E5 in general for inoculants, but understanding that there's a lower amount there is important compared to the bradyrhizobium. And then the last thing is to understand how to store and use the product in terms of application. General rules of thumb with any living microbe product, some require refrigeration. It should say clearly on the label if refrigeration is needed. They might just not have a good temperature tolerance, and refrigeration will help with the shelf life. But most of these products will do okay. If they don't need refrigeration, they'll do okay at room temperature. What you shouldn't do is take them out and put them in your truck in the sunshine and under hot conditions, because that's how the microbes are going to die, okay? 70 degrees or so, room office conditions, fine. But, 90, 100 degrees, not so good for these living products, okay? And then make sure that before you use the product, you shake it real good. A lot of times these, especially, for a liquid product, they'll settle out, so you do want to shake it a little bit and make sure everything's back in suspension when you pour it out of the container. Okay. The importance of a high concentration and high viability when you go out and deliver these products. I can't emphasize this enough. And the reason for this is these microbes are going to have to go into a different environment in the soil. And what happens is they're going to experience potentially hostile conditions. There could be other microbes there that they have to compete for resources. Temperature, moisture, pH, what's happening in the soil is important. And then time, because as time goes on, if they don't have adequate nutrients, and there may be nutrients, in the carrier that you're using to apply the microbes or there in the soil solution, the more that you can kind of help protect them and give them a good environment, the better. Because over time, as we're waiting on the seed to germinate, which hopefully doesn't take too much time, they're going to lose count, right? We're going to lose some logs potentially. And so what we want is we want enough to survive and proliferate in that soil environment that they're going to start to colonize the root system. And that's where that direct relationship with the plant occurs, okay? And what this looks like, at least in a lab setting, is some microbes are very good at colonizing the root system. As an example, Pseudomonas is known as a type of microbe that is. And what will happen is as that root system emerges, the plants will be fed by the root exudates, and then they'll continue to grow with the system, okay? Real quick on delivery, seed treatment, with biologicals, the more precise the delivery, the better, because some of these products can be fairly expensive, right? So precision delivery is important. And so seed treatment for convenience, I know a lot of growers like to use seed treatments for that reason, but to understand what's happening to the microbe, it's important, when you deliver a liquid as a seed treatment, which a lot of these seed treaters are, the microbe, that liquid during that seed treatment process is going to dry down, and microbes do not like the desiccation process. That dry down, you will lose potentially multiple logs of microbes through that process, because during seed treatment it's very uncontrolled. Okay, so that is one aspect you have to be aware of with using a microbe in a seed treater. Dry formulations of microbes are becoming more popular. Part of the reason for this is we can create a dry formulation in the lab, commercially, using very controlled processes. So we can control what the microbe experiences. So that desiccation process we can manage. And then they hang out in this dormant state for a period of time, which is good. They're not actively metabolizing and potentially dying. They're just kind of hanging out. So that by the time we treat them as a dry on seed, they continue to, as long as that seed can remain in that state, they can continue to kind of exist without dying to the same extent as a liquid application does. Liquid in furrow is another common method for delivery of biologicals. The nice part about that is you have more flexibility. The trade-off is with a tank mix, whatever you're putting the microbes in, they're going to be in contact with something else. And if that's a chemistry or even water, it can be problematic for the microbes. So something to keep in mind is what's in your tank mix. Things to be aware of with starter fertilizers, compatibility. I think I might get into that in the next slide. Yeah, I do. Compatibility with salt index of the salt. I'll talk about that in the next slide. A lot of times I have people ask me about 2x2 application for microbes. And 2x2 for microbes is not, I don't recommend it, because now you're putting the microbes away from where the seed is, so they have to wait longer till they really start to be able to associate with the plant's root system. Other biologicals, like I talked about, organic acids, amino acids, and so forth, are fine, can be fine as a part of a 2x2 application. So you just need to understand from the product's label what's a preferred application method. But for microbes, I would not recommend it. Foliar, you're going to see some more products that are going to be recommended to be delivered by foliar. I mentioned some of those nitrogen-fixing products that are endophytes that get into the plant cell. They actually can provide a lot of benefit, particularly in corn, in later stages of growth when the corn plant gets really needy for nitrogen. If you can have a nitrogen fixer you're applying at a later stage foliar, now you're able to fix nitrogen at those later stages. So that's pretty powerful and impactful, and you're starting to see some products on the market that can do that. All right, real quick, getting back to compatibility, things to be aware of with microbes, salt index of a starter fertilizer, keep it less than 20. 1034.0, I know you guys love to use it, but it can be really bad for microbes. It's just too hot. Can be too hot. pH, typically, if you have lower pHs down in the 4.5 range, it can be detrimental to microbes. More neutral pHs are better, closer to 7. Be mindful of any other micronutrients or fungicides, insecticides you might be tank mixing with or applying as a seed treatment, as they may not play well with the microbes. All right, quick story here. Last year we did field trials, and we had 4 different, these were production acres. We had 4 different farmers involved in this study, and I asked them to send me, we do a lot of compatibility testing in our labs, and I asked them to send me samples, and I put together this nice kit with file and labels and packaging and stuff, and one of the growers sent back a Coke bottle with the starter in it. My lab manager kind of freaked out. I thought it was funny. But I know you guys are busy at planting time. No matter how the sample gets to me, I'm not gonna, even if it's in a Coke bottle, it's fine. All right, something to remember here, I'm gonna show you data, a lot of data real quick of log loss. So all these numbers represent log loss. What we did was we looked at compatibility of the microbes with these different, basically tank mix components. Most of them are starter fertilizers, and compared it to a buffer control. And anywhere where you see kind of the yellow and ones and close to 1, these are log losses. And as you remember, a log loss equals 90%. We're losing 90% or more of the microbes just by sitting in that tank mix solution for a period of time. And we were looking at 3 different strains as part of this study. What was surprising to me, you can see the 1034.0 there in the middle. And we did for at least 2 out of the 3 strains, we saw some impact right away. What I wasn't expecting over at the end, you can see the water, locally sourced water. We actually saw an impact from water that we got from one of the sites. Water, whether it's from city supply or well water, can contain, whether it's chlorine or minerals that can impact microbes. And actually microbes in general do better in a slightly salty solution, a saline solution that's a buffer, than pure water. So we did see an impact initially with just water, so something to be mindful of. And as time went on, a lot of those effects became worse. In some cases, we were losing 2 or 3 logs. The key here is, if you're going to do a tank mix and include microbes, make sure the microbes are compatible with what you're mixing with, and don't let it sit for overnight or for the next day. Use it all the same day, preferably within a couple hours. Log loss happens on seed as well as part of seed treatments. I talked about this earlier with just the dry down impact of the microbes going through drying and desiccation. But then over time, whether it's the same day, so this data comes from a study where they looked at 3 hours and then 30 days, and they looked at, this was a Brady rhizobium soybean inoculant being applied as a seed treatment with 3 different different chemistry packages as part of the seed treatment. And you can see that you see multiple log losses with different chemistries being used, but then that impact over time, you can see multiple log losses. So any time those microbes are sitting there for days and weeks, potentially months, you're seeing loss of those microbes, okay? That impacts efficacy in the field, bottom line. So be aware of that. All right, the last bit here I'm gonna go through, hopefully it'll be useful in terms of where you can go to get information, okay, on how these products work. And I will say, this is an example that comes from work that Ohio State, this is Dr. Laura Lindsay's lab, did a couple years ago. The bottom product there, BioYield, is one that 3Bar had in the study that we produce. And the rest of the products are soybean inoculants, so Brady rhizobium strains for the most part. Some included a couple different strains. The thing that you want to see in studies, and these were done at a university on replicated small plots, which a lot of the university trials are done that way, not on production acres. But what you want to see is multiple years, and in this case, there's a focus on yield response. And what you want to see is there consistency year to year? How can you compare it back to the untreated control? And in this case, they were looking at a number of different products. So you can get an idea of what these products, what the yield response was, okay? So good university extension should be a good source for data. More and more universities are producing this kind of data, including Ohio State. Another great resource, we're seeing more, I would say, downstream, whether it's a distributor or manufacturer, putting data out there. And I think Beck's does an excellent job with their PFR proven resource, and they've, over the last 5 or 6 years, been looking at different biological products. So I took all the data on biologicals that they have out there that have been proven as part of their process, and you can see at the top what each product is, starting with a nitrogen fixer, so an azospirillum being used on corn, Brady rhizobium being used on soybean, and you can see some others that involve extracts and sugar and humic. And then to finish it off here, some other products that they did more recently. The nice thing that they also do is they provide each year's data. So you want to look at over multiple years. Are you seeing, fairly consistent response? They do their studies over multiple sites, which is nice. And then the other thing that can be useful is you can go to the manufacturer's website. In this case, the Terracin product, as part of Beck's study, they looked at between 2020 and 2022, they did field trials. On New Leaf, who's the manufacturer of this product, they published more data, and their data comes, I think, from the years of 2017 to 2019. But you can see, was there consistency year over year, multiple locations, and you can start to dig in and understand. And that's what you're really looking for. Will it work on your acres? I don't know. But you're looking for at least some signs from research that it's working consistently in some other places, okay? And having third parties involved is obviously seeing that there versus a manufacturer's website is obviously helpful as well. Okay, so these are good resources to start to dig into some of these different products. And what I would say with getting here at the end, looks like I'll have time for questions too, which is great. A summary of best practices. Make sure you, like I said before, make sure you understand the label. Does it have a high concentration of CFUs per mil or CFUs per gram to start? That's a good place. You want a lot there if they're going to do anything. A good target is to try to achieve 10 to the 5 to 10 to the 6 CFUs per seed, okay? To know that you have enough there even after delivery. Store your products under appropriate conditions. Room temperature should be fine, but if it says refrigerate, by all means refrigerate, because it's not meant to survive at room temperature. Keep them out of your trucks on hot days. In terms of delivery, seed treatment, some things to keep in mind. If possible, plant the same day as you treat, and you're looking at me like, no, that doesn't happen on my farm. But understand that the longer you wait to plant, you are going to lose some microbes from that seed treatment. Dry application may protect the microbes better for longer. I can't wait till I have data that I can support this hypothesis, but I do believe that dry, if you keep those microbes dry, they're going to do better by the time they reach the field. You're not putting them through a dry down as part of a liquid seed treatment. That's my personal opinion right now, and I'm looking forward to having some data to support that here soon. In-furrow delivery is an alternative to seed treatment. Make sure, if you're using a starter fertilizer, it's a low salt index fertilizer. Anything else you're tank mixing, including water, try to understand if there's compatibility. And consider that there are new technologies out there. Again, I didn't get into those delivery technologies here in this presentation, but I did talk a lot about them in the presentation I gave this morning, so you can check that out. And this is the last slide, where to start, okay? And these are just recommendations, I think. I know you guys are faced with a lot of product choices. You're trying to figure it out. I'm in the industry, and I'm trying to figure it out, okay? So there's a lot to learn. We're really at the beginning of this journey, I think. And so where to start? Define what you need to achieve, right? What do you need? What do you want to achieve? Maybe it is nitrogen fixation, right? You're trying to see if that makes an impact. Maybe you want to reduce your nitrogen application and want to use a biological to help with that. Maybe it's you want to mitigate certain stresses. Maybe you live someplace where you have early emergence problems because it's cold and wet and you want to get out there earlier, right? There are microbes that can help with that. Or maybe your conditions are too dry. So think about what you need first. And then you need to find a product and understand what it does to meet that need. Don't just use something because your neighbor used it, right? Understand what you need and make sure you're using a product that can meet that need, okay? And understand how it works. And this is where it gets, I understand, there's a lot of information out there, and trying to understand it can be hard. Consider a zone, start with just one field or one zone, right? And particularly if you have a lower yielding field, this could be useful to use biological in that lower yielding area. So pick one zone and start there. And then consider your best application method. It has to fit in with your practices, but I provided some information here today. Consider the best method based on what the microbe is and your practices. Remember that not all the value comes from yield. Yield's what we're trying to achieve, right, and increases in yield, but it can come back to ROI, it can come back to early vigor or better seed emergence. Think about it beyond just increasing yield in the use of these products. And then try at least 2 years. More than 2 years is even better, but at least 2 years. Don't give up after the first year, okay? And that is it.
Hi, you touched on some of the loss of logs.
Yeah.
Can you restore those? Can you add some microbes back into that treatment and get that all replenished on your farm?
Yeah, so when you receive a product as an input, right, that loss is going to be contained in that jug, right? When you apply it in the field, there will be the ability to multiply and proliferate with the plant. So it will do its job in the field if there's enough there and they survive through delivery, right? From somebody in the industry, don't open the jug and try to mess with it, I guess is my answer to your question. If you add more sugar and they have food, microbes will grow. And with a liquid formulation, the thing you have to understand is, you receive a jug that has microbes in it. Those microbes were grown at the manufacturer. They added as part of their formulation things to keep the microbes happy because they're actively metabolizing in that jug because they're in liquid and they need nutrients. If they run out of nutrients, they die, okay, or they'll start to die. And so if you add more, the challenge is you don't want any contaminants to get in there from the air, because they'll grow too. And now you have a compromised product that may not be very effective because you have other things in there. Okay, does that make sense?
Well, you kind of touched on things happen on the farm where we're mixing things and all of a sudden something comes up. We leave it set for a couple hours to a day.
Yep.
Well, you're losing your logs. Can we add something to it at that point in time to recharge them?
You could, but as it's open to the environment, you're compromising. If you add more to it, anything that kind of gets in there or may already be there is going to grow too. So be careful. You could, but I would not recommend it.
I kind of see a PR nightmare coming with this when the non-farmers hear that we're putting live bacteria on your seed or modified bacteria on your seed. As your companies, are they going to do a better job than they did with, say, the Roundup Ready fiasco?
I sure hope so. I mean, I think synthetic biology is a fancy term, right? I think there's definitely lessons learned from the Monsanto days, right? We're dealing with different than crop traits, right? We're dealing with microbes. I think what you're going to see is this coming together and utilizing synthetic biology to help both crop traits and the biologicals that are being matched with them, these microbes, to a point where we see, this is where, when we talk about moving the needle in the next, biotech revolution, green revolution, it's going to come through these high-tech types of advancements. How it's handled from a PR perspective, I am not a PR expert. I hope we do better. But this is where, the R&D is going. And it won't just be with the microbes. It really is going to involve both, it's matchmaking and being able to use synthetic biology to make it work better, right? And that's where we're going to see this next jump in our productivity.
Question here. I'm trying to understand, so is this the microbial activity, is it built around proliferation? In other words, what comes out of the jug, the expectation is it's going to exponentially multiply? Question one. And question two is can things like heavy rain events where the soils go anaerobic, can that completely wipe out that colonization at that point?
Yeah, great questions. A lot's happening. I hate to say it depends, right? But key to delivery is being able to have a high concentration there. So statistically, even if we lose some, we're still going to be okay. And that time frame where before the seed germinates, if there's not food there, you'll see some loss there. That proliferation really happens if there's enough food. And what happens when there's some microbes as part of nutrient cycling, but what is happening with the plant, some of these microbes will get inside the plant and there's a cycling process. So if we can get to that point and root colonization happens, they are going to multiply and proliferate with the plant, okay? But it's all tied to the plant. It's not impacting the broader microbiome in the soil, only right there in the rhizosphere of the soil environment. When you have events like rain and things, you know, the microbes that, especially the endophytic ones that can get inside the plant, you know, that's what they're trying to do, and they'll be fine. But differences in weather and things happening can impact. Most of these microbes that exist in the soil are not purely aerobic. A lot of them are known as facultative anaerobes, which is a fancy way of saying they can exist if there's not any oxygen there or low levels of oxygen, or they can go either way. And that's what a lot of these soil microbes are. So they can handle it in some sense because they've evolved to be able to handle it. Different from a pure aerobic microbe or a pure anaerobic microbe that can't handle, either needs oxygen or can't handle oxygen. So they fall in this middle space, actually. Oops.